So now I'd like to introduce our next speaker. Amanda Guth was born in Pennsylvania. She is an undergraduate of the University of New Mexico and then went on to get her graduate degree, which is a PhD in immunology from the University of Colorado Health Science Center. She went to vet school right here in Colorado State and then stayed on after that as a postdoc and then became an assistant professor. She is she's studying tumor microenvironment, developing better ways to develop immune therapies, and that includes cancer vaccine, which is what she's going to talk about today. She co-founded Photon Pharma with Ray Goodrich in 2008, and they are doing some really exciting things. So, Amanda?
I'd like to thank all the sponsors of NoCo Biosciences for allowing me to talk today about our CSU startup company, Photon Pharma. So a lot of the slides that Heidi presented, we have been through some of those processes and understand kind of the challenges, and are really excited that CSU is pushing forward with having us be able to develop these startup companies and be successful. So today I'm going to talk about our product, which is a novel autologous cancer vaccine, and we're looking to target both pets and humans with this vaccine. So we all know that traditional cancer therapies that have shown some success consist of mostly chemotherapy and radiation therapy. In the last few years, cancer immunotherapy has sort of been in the forefront as people really learned more about how cancer and the immune system work together. And so nowadays, you can see that cancer immunotherapies have become part of standard of care. So we are developing a cancer therapy that is a cell-based cancer therapy, and kind of a similar product to ours that has been in therapeutic use is known as CAR T cells. So not to really get heavy into science, but CAR T cells are basically where people take blood cells out of a patient. They engineer their immune cells to be specific to a tumor type that patient has, and they put those cells back in, and those cells then go off and fight the cancer. This has shown great success in what we call liquid or blood cancer, so lymphoma or leukemias, because these cells have a single antigen that the cells that are pulled out and engineered to target can target, and it doesn't matter if you kill off every cell that has this antigen in it. So this limits the therapy of these T cells right now to mostly liquid cancers when you're looking at lymphoma and leukemia, where you can take out all of your B cells or all of your T cells. Interestingly, there's a large turnaround time with this therapy, about 17 days on a good day from vein to vein clinical trials, because there's a lot of manipulation that's needed. It's also extremely expensive, and this has been a huge talking point amongst people in industry and in medical centers. Right now, the cost of this therapy is almost $500,000, but it does offer the success that you can potentially get a cure from it. So we were interested in developing sort of a modified version of this. We call it the reverse CAR T cell therapy, and our product is also known as the in-a-cell vaccine. And our company is developing this vaccine based on some riboflavin and UV light technology that allows us to inactivate the cells. So how is our process different from previous cancer vaccines that people have tested? So previously, when people have taken tumor cells out of a patient and inactivated them, they've used methods such as gamma radiation or UV irradiation that were done at high doses or even involved some heat treatment. And we believe that this actually disrupts a lot of the surface antigens that are necessary to stimulate the immune system. So in our case, we use a low level of UV light and riboflavin that can target the DNA, and this process inactivates those tumor cells that they can't replicate, but still maintains the integrity of these proteins on the surface, which can serve when you put it back in the patient to stimulate their own immune response against the tumor. How this process works is shown here, where you have your cell in solution with riboflavin. The riboflavin binds to the DNA, and then a certain wavelength of light energy, there's an electron transfer that then disrupts the nucleic acids of the cells, and therefore the DNA is unable to replicate. But the proteins are still remained intact in these cells. So this is our process that we've developed, where we get tumor tissue, whether it's from a biopsy or from a surgical procedure from a patient. We add riboflavin solution to those cells after they're put into single cell suspension. We do a light inactivation treatment, which only involves about 2 to 3 minutes of time, so it's a very fast procedure. And then afterwards, we take those inactivated cells and make multiple aliquots that eventually will go into development of vaccines for the patient. So in this case, we're being able to take a patient's own tumor and develop a personalized vaccine. The advantage of our technology is we actually can do this same day. So we can have a patient come in the morning, get a tumor biopsy from that patient, and have a vaccine ready for them in the afternoon. It's also very low cost. Riboflavin is relatively cheap, and the process is pretty simple. So our technology here then is a live replication inactivated tumor cell vaccine. And this here again kind of shows our process, showing how we can go from lab to a tumor biopsy to the lab to inoculants. The machine we're currently using is one from Terumo BCT. And the beauty of these machines is they're pretty small. And so we envision in the future that potentially this therapy could be available in almost every hospital across the country. Interestingly, and specifically including rural hospitals that may not have access to other personalized therapies, more of the CAR T cell therapy where you have to send samples out to get processed and then ship them back. Our process may actually be able to be done in-house. So what evidence do we have that our vaccine process will work? So I'm just going to present some of our sort of preclinical studies we've done, and we'll go from there. So first thing we needed to show is that we are actually inactivating the tumor cells. So we have done this across multiple different species. We've done mouse, human, and dog cells. I'm not going to go into specifics of these slides. They're kind of hard to see anyway, but to suffice to say that with all the antigens we looked at when we inactivate these cells, when we looked at different surface proteins, all these are maintained to some level. So we don't seem to be losing any proteins when we do this inactivation process. And in fact, some of the data we've generated when we've looked at known tumor-specific or tumor-associated antigens that belong to certain tumor types, in some cases we actually see upregulation of those tumor antigens. So next, we wanted to, of course, show that it's safe. So we did this multiple ways. So when we treated tumor cells from a tumor that was extracted from a mouse, when we look at the cell lines, which is considered the live there, you can see that at 24 and 72 hours, the cells are proliferating as they should. When we take tumors out of a mouse, which is the ex vivo one, same thing at 24 and 72 hours, those cells are still proliferating. When we take those same tumor cells out of the mouse and do our inactivation process, there is no proliferation noted at 24 hours and 72 hours. The middle slide shows a human colon carcinoma cell line. And this is just using a microscopic dye that we can look at to see if the cells are proliferating. And again, the inactivated cells do not pick up this dye, which shows DNA that's dividing. Lastly, we've looked at both immune sufficient and immune deficient mice. So we've taken tumors out, inactivated them, and put them into these mice. And we followed these mice out for quite a few months and did not see regrowth of the tumor. So we feel our process is safe and that we are completely inactivating these tumor cells. Next, we want to look and see, are we actually triggering immune responses? So we did a couple of different assays. Again, I'm not going to go into great detail about them. Suffice to say that we are able to stimulate T cells in mice against tumor antigens associated with the inactivated tumor cells. The middle panel shows antibody responses, so we're able to induce a humoral immune response against these inactivated cells. And then lastly, it shows activated dendritic cells, so we're able to stimulate the antigen-presenting cell component of mice that are infected with tumors. And this is important because these cells need to present the foreign tumor antigens to the immune system to get them stimulated. So it shows that our vaccine is actually triggering pretty much all the arms of the immune system. So we initially started in some rodent models. So this is a rodent mouse model of primary breast cancer. And we show in the corner, the top corner there, that when we treat mice with in-a-cell vaccine once a week, we do get reduction in tumor growth. And this is shown related to the decrease in doubling size of the tumor, kind of shown over on the far side there. And then lastly, the survival of these mice is increased when they just received the vaccine. Next, we tested this in a metastatic disease model, and this model was pretty neat. This is where I got to use my veterinary skills, although I did learn that I'm glad I did not go into veterinary surgery. So we gave mice tumors, primary breast cancer tumors, And after they reached a certain size, we surgically removed the tumors from these mice. The mice were not euthanized at the time. Their tumor tissue was collected. Then these same mice were able to receive vaccines of their own tumor. So we basically mocked what we would do in veterinary or human patients using this mouse model. These tumor cells express a enzyme called luciferase, and if you give it the substrate, you can actually measure the amount of tumor cells using an imaging device, and that's kind of shown. No, I guess it didn't show up. Interesting. Oh, no, so my next slide, sorry. It's photon flux, so it can measure the flux of the luciferin that's being catalyzed. And what we can do is use it as a measurement of metastatic disease, and this allows us to follow individual mice over time. And so what we saw is the mice that received the vaccine as compared to control mice or mice that received just our adjuvant system actually had a reduction in the development of metastatic disease. And here's just showing some mice at a time point. This is, I think, a 14-day time point, where you can see here where the imaging is of a control mouse. Again, those colors correspond to tumor burden. And then the Inacel treated mouse at that same time did not have any evidence of metastatic disease. So we believe the mechanism of action of our vaccine is we are changing the tumor microenvironment. So there are groups of T cells that are associated with immune suppression in the tumor microenvironment. And if you guys have heard about immune checkpoint inhibitors, those chemicals are involved in actually overcoming this immune suppression by blocking the receptor that causes cell-mediated or T cells to not respond to the tumor. What we found with our vaccine is we're actually able to modify those cells as well. So we see a decrease in T regulatory cells, which is a population of cells that dampen the immune response, as well as changes in expression of known tumor-associated or T cell-associated exhaustion molecules. So these are molecules that are expressed that cause the immune system to sort of shut down. So we feel that our vaccine is having an effect in reducing tumor burden by modifying the tumor microenvironment. And this is big, and this is a really big, interesting area in cancer research right now, because with the immune checkpoint molecules, if you can demonstrate a therapy that will increase the amount of immune cells that are in the tumor that are active, what we call sort of a hot tumor, it makes it more amenable to a lot of these other immunotherapies. And that's sort of how we envision our therapy moving forward, is it may not necessarily be a standalone therapy, but it can be combined with other immune therapies out there to allow tumors that may be immunologically not quite as hot to become more hotter and more receptive to these other therapies. So here are our future plans. So we're continuing some preclinical studies, likely focusing on triple-negative breast cancer as sort of our first in-human patients. We're doing some studies now to compare our inactivation technology to what's been done before, in particular gamma radiation. We're developing our adjuvant system, sort of optimizing what adjuvant system we want to combine our vaccine with, as well as looking at dose and then also characterizing the immune response a little bit more. We have begun one clinical trial using some canine patients at the CSU Flint Animal Cancer Center. So as Heidi alluded to, it's great right here at CSU where we have access to really moving our technology into veterinary patients rather rapidly. And that gives us, again, I'm on the same board she is, where, you know, these mouse models are great, but you have to study these processes and these treatments in animals that naturally develop the disease in their outbred populations. We're going to begin hopefully working with Biomark to start to develop some GMP production processes using patient samples, initially probably using dog tissue samples, which is, again, a great resource we have here. And then talk about sort of commercialization model and production center setup. We're also in the process of designing our human clinical trial, and this includes developing our IND package. We're about to submit an Interact package, which is like a pre-IND package, in conjunction with a group called Cancer Insight, who are helping us design this. So here's just briefly about our canine proof of concept study. So we have enrolled 2 dogs in this study, and this is just to test that we can do this process, that it works, and that there is no adverse events happening in the dogs. So this is our first patient, Ella, and Ella's owner gave us permission to show her the picture here of her on the end. And this is basically the process. So we get tumor biopsy sample or tumor surgical sample. In this case, Ella had liver hepatocellular carcinoma. So we got some liver tissue sample. We mix in riboflavin, which is the yellow solution there, and then we put it into the illuminator device in the middle. It goes for about 1 minute and 30 seconds, about 2 minutes, and that inactivates the cell, and then we make separate little aliquots of that, and then the patient gets vaccinated. So, and Ella had a febrile response about 8 hours after vaccination, which for immunologists is a very good sign because that means your immune system is actually responding to the vaccine. She recovered from that with no ill effects. It was a little lethargic the next day. Again, sort of in our mind is actually good news because that shows that the immune system is working to respond against that vaccine. We've enrolled a second patient, and that patient as well had no adverse events that were associated with giving this vaccine. Our plan is to do one more patient just to verify, and then we'll at least have our kind of safety data to move forward with this. This is our developmental timeline here. So again, we're sort of the dog safety study. We're going to start looking at in vitro human cell lines. We're looking to move forward with a bigger dog trial. So once we get sort of our standard operating procedures and our manufacturing procedures nailed down, we're going to open a larger dog trial where we can look at efficacy. And again, kind of on the sidelines here, we're working on some hardware, software, and disposable development to make our product. So this is our Photon Pharma team, the 3 co-founders. So Ray Goodrich, who will talk later today, is here, as well as John Weston, who's our CEO. I unfortunately have to run after this. My son has a soccer tournament, and I need to get him to that. But both Ray and John will be around if anybody has any questions. And so one of the new things we do with some of our students and posters is this cool QR code thing. I don't know if it'll work from the audience, but if you use your phone on this QR code, it'll take you to our website, and it's listed here as well. So with that, I'll take any questions. Thank you.